All right, well, thank you so much for the invitation to present my work to you all today. I've been in the sleep field now for over 20 years, and most of my time has actually been spent investigating the neural circuits involved in regulating sleep and that are influenced by general anesthetics. And I looked from the role of adenosine and acetylcholine in sleep control. I've used arousal circuits to try and reverse anesthesia. But I've always been personally interested in metabolism and healthy aging. And so now I've come to a point where I can integrate my personal interests in healthy aging and my professional interests in sleep in order to study the intersection of metabolism and sleep in health and disease. So the central theme of my lab is looking at how metabolic and sleep interventions can be involved to influence brain health. And the way I've done that is I've focused on 4 different topics. So first, the disrupted sleep with aging, and then postoperative delirium. And now we're expanding into Alzheimer's disease, as Mike mentioned, and mental health. And so you may think, well, what are the common deficits in all these different disorders? And it turns out that both metabolism and sleep influence a lot of these different diseases. And so it can be common applications can be used to address each of these. So why do we care about metabolism? Poor body metabolism influences brain metabolism, and the metabolic health in the country is getting worse. A good measure of metabolic health is metabolic syndrome, and metabolic syndrome is characterized as having 3 of the following criteria: abdominal obesity, high blood pressure, triglycerides are high, low HDL cholesterol, or high insulin resistance. So metabolic syndrome is a problem because it increases your risk of cardiovascular disease, diabetes, stroke, and other neurological complications. And remarkably, 1 in 3 adults in the US has metabolic syndrome, and over half of the adults are either diabetic or pre-diabetic in the US. And a good marker of metabolic syndrome is obesity, and obesity has actually been tracked in the US for many years. Here's a nice graph that shows how striking the difference of obesity has become in the last 30 years. You can see in the left in blue that in 1990, 30 years ago, it was only 10 to 14% of the population. And now it's up to even 45% of the population. And many parts are in the 30% or more in the US. In addition, metabolic syndrome gets worse with age. So what do we know about metabolism and brain health? So all cells in the body need energy to generate ATP. And the brain, while it's only 2% of your body weight, uses up to 20% of our energy. And so with this brain's high energy demand, it needs a continuous supply to maintain consciousness. And interestingly, our brain is like a hybrid car. It can work on either glucose or on ketones. And ketones are generated by fatty acids and get more energy with less oxidative stress and less metabolic waste in fewer steps than glucose. So what are ketones? There's 3 main ketones: acetone, acetoacetate, and beta-hydroxybutyrate. They're naturally produced from the liver, from fatty acids during exercise, fasting, or ketogenic diets or ketone supplements. They can be readily used by the brain, heart, and skeletal muscle. And in addition to being a fuel source, they have signaling activity and they decrease oxidative stress and are anti-inflammatory. So you may have heard of ketoacidosis. That's diabetic ketoacidosis occurs at a range of 10 millimolar. The levels that we're talking about for therapeutic purposes are more in the range of 1 to 3 millimolar. So it's an order of magnitude below ketoacidosis. Why are ketones important evolutionarily? Well, historically, food was scarce, and humans went through regular periods of fasting to be able, the ability to store energy as fat was very advantageous. Only our ancestors that could survive with their own fat were able to keep going and reproduce. And so in addition, ketogenesis helped fuel enlargement of the human brain during its evolution. And today's food environment, there's an overabundance of food, but this ability to store fat is less helpful, but we can use the ability of ketones as fuel to our advantage. In fact, Even ketogenic metabolic therapies have been known for over a century, back to the 5th century BC, where Hippocrates reported that complete abstinence from food and drink was an effective cure for a man with epilepsy. And then in the 1920s, the ketogenic diet was developed at Johns Hopkins as a fasting mimicking diet for pediatric epilepsy. Obviously, you can't not eat forever, so you needed something that could reduce seizures, but still have the beneficial effect of reducing seizures and eating. And so the ketogenic diet decreases seizures with many different etiologies. And for some people, it completely cures them. It's actually pretty remarkable that patients, especially pediatric patients, can go on the diet for 2 to 5 years and then come off of it and remain seizure-free and drug-free for the rest of their life. So what else changes with the brain and metabolism with age? So you have mitochondrial dysfunction, there's decreased glucose uptake and insulin resistance, you have increased oxidative stress and inflammation, and a unique feature of the older brain is that its ability to use this glucose declines, but its ability to use ketones remains. And we can see this in PET imaging of people who are either controlled, healthy, mild cognitive impairment, or with Alzheimer's disease. And here, this is PET imaging for glucose. And you can see how the amount of glucose used goes down as the cognitive function gets worse. And then this is for acetoacetate, one of those ketones. And you can see that even in the Alzheimer's brain, there is still robust uptake of ketones. So these data demonstrate that this deterioration in brain energy metabolism with MCI and Alzheimer's disease is specific to glucose. So this older brain's ability to use ketones provides therapeutic potential. So brain metabolism, brain metabolic dysregulation affects many neurological problems, including sleep disruption, this postoperative delirium, neurodegenerative diseases like Alzheimer's disease and Parkinson's disease. And then another area I'll mention at the end is mental health, like anxiety, bipolar disorder, schizophrenia, and major depression, as well as the well-studied epilepsy, autism, and many cancers, including glioblastoma. So I've focused on the first 4 of these for my research lab. So as many of you know, sleep is disrupted with aging, and both non-REM and REM sleep decline, and especially the deep slow wave sleep. Sleep becomes more fragmented. Circadian rhythms change. There's phase advances and a decreased ability to adjust to phase shifts and reduced amplitude of circadian rhythms. In addition to the disrupted sleep, there's also glucose inhibition and insulin resistance and alterations in appetite hormones. So what ties together sleep, metabolism, and aging? Well, it turns out that oxidative stress links energy metabolism to sleep and aging. So oxidative stress is the imbalance of reactive oxygen species generated by metabolic processes and the balance with antioxidant responses. Oxidative stress can damage lipids, proteins, and DNA, and it's associated with aging. There's a bidirectional relationship between sleep and oxidative stress, where sleep loss increases reactive oxygen species, and then the oxidative stress in turn triggers sleep. So ketones improve mitochondrial function and reduce oxidative stress. They increase antioxidants and they scavenge free radicals. So they could be useful in this oxidative stress dysregulation with aging. Therefore, we've tested the ability of ketones to improve the disrupted sleep with aging via this improved brain energy metabolism and decreased oxidative stress. And in fact, there's some studies that suggest that this might be the case. This is a study in rodents looking at slow wave activity over time after a central injection of acetoacetate, that's one of the ketone bodies, and looking at how much slow wave activity there was in the sleep following that injection. And you can see that with higher levels of acetoacetate, they had higher levels of slow wave activity. So that suggests ketones could be a promising approach to improve sleep quality. Now in humans, I collaborated with a company that's using nutritional ketosis to reverse diabetes. And they had patients with either type 2 diabetes on their ketogenic diet program, people with prediabetes, or people just on the standard of care, usual care type 2 diabetes. And you can see using the Pittsburgh Sleep Quality Index that patients that were receiving their ketogenic intervention had better improvement in their overall global sleep score, their subjective sleep quality, their sleep disturbance, and their daytime dysfunction. So this suggests that ketogenic diets could improve sleep quality. So what do we know about the changes in metabolism across sleep and wakefulness? This is from a nice review looking at the differences in blood versus brain glucose, lactate, fatty acid, and ketone body levels across sleep and wakefulness. So interestingly, during sleep, blood glucose goes down because you're not eating anymore, whereas brain glucose actually goes up. And so this has been shown across sleep and wakefulness, and we wanted to show that as well, and then expand that to anesthesia. So what did we do? We used a biosensor for glucose or lactate, which involves having a semi-permeable membrane and an enzyme specific to your analyte of interest, which then generates hydrogen peroxide and causes a potential on a platinum electrode. And so in order to do this, you have to put in the probe, it acclimates for a little bit, and then we were able to record sleep and animals under sevoflurane anesthesia in the same experiment and look at changes in sleep, in glucose and lactate levels across both sleep, wake, and the anesthetic state from sevoflurane. So this shows prefrontal cortex, glucose and lactate levels, glucose in blue and lactate in red, across the sleep-wake cycle. A standard hypnogram. Here we have wake and non-REM. You can see that during wakefulness, lactate levels go up and glucose levels go down. And then the opposite is true during non-REM sleep. The glucose levels go up in the brain and the lactate levels go down. And so we thought this was quite interesting. It's consistent with some previous data, but we wanted to compare how that was related to what happened during anesthesia. So if we looked at the anesthetic period, this is 3% sevoflurane anesthesia. You can see that there's a transition period here where we have a similar pattern to wake. And then once the animal gets into a deeper state, this is the spectrogram looking at lots of delta power, meaning they're in deeper anesthesia, we get a similar pattern to what we saw during non-REM sleep, where there's low lactate and high glucose. And then when the animal wakes up, we have the opposite, and we see the high lactate and the low glucose. And we quantified this across both sleep and wake and sevoflurane anesthesia. So you can look at the spectrogram on the top, the hypnogram next, and then the amount of lactate across these transitions between wake and non-REM sleep versus sevoflurane anesthesia during this induction period and during deep anesthesia. And this is on the same scale, so you can see the amount of fluctuation during non-REM sleep is much smaller than the amount of fluctuation during sevoflurane. So we're now going to move on to the second portion of my talk, talking about postoperative delirium. So postoperative delirium is an acute syndrome characterized by inattention, disorganized thinking, and altered levels of consciousness, and it occurs more often in patients over 65. So consequences of postoperative delirium include prolonged ICU stay, long-term neurocognitive deficits, and increased mortality. And so while the neuropathogenesis of delirium is incompletely understood, there's factors that contribute, including age as a large one, inflammation, vascular dysfunction, disrupted brain metabolism, sleep disruption, neurotransmitter imbalance, and impaired network connectivity. So if we look at this over time, we can see that there are predisposing factors on the spectrum of aging. So some people of the same age have different sort of fragility levels, and some are healthier. And they have different levels of inflammaging, that's the amount of inflammation with aging, vascular changes, oxidative stress and body composition, and then different neuroendocrine functions and neuroanatomical abnormalities. So the combination of this predisposing factors with the acute stress of surgery can lead to delirium, where there's these vascular inflammatory problems and physical and psychological stress that can lead to poor health outcomes from having this postoperative delirium, including disability, institutionalization, hospitalization, and death. So how does sleep contribute to delirium? So poor sleep quality and delirium are common, and they often coexist. Sleep quality declines with age, and the probability of delirium increases with age. There's disrupted sleep before surgery can lead to increased risk of delirium, as well as postoperative sleep disruption. Sleep and circadian rhythm disruption from drugs and the lights and the hospital interactions in the ICU can also contribute to an increased risk of delirium. What about metabolic contributions to delirium? So metabolic syndrome has been shown to be significantly increase the risk of postoperative delirium. That's the metabolic syndrome that we were just talking about. 1 in 3 adults has in the United States. And so you can see that's a big burden for the hospital population. Acute surgery also causes inflammation and disrupts energy metabolism, making the older person more vulnerable and likely to develop delirium. Metabolomic approaches have shown that there's also differences in lipid metabolism with people who develop delirium or people who do not. And then also related to oxidative stress capacity has been associated with postoperative delirium development. So what does that mean for our studies and the likelihood of metabolic interventions for burst suppression? So first we have to define what is burst suppression. Burst suppression is shown here in this diagram on top, where there's a period of bursts and suppressions compared to a regular EEG that many of you have probably seen. This occurs during deep anesthesia, sometimes during hypothermia and other anatomical conditions. So the duration of burst suppression during an operation correlates with the likelihood of developing delirium, but interventional studies have had mixed results. And that may be because burst suppression is an indicator of the underlying metabolic health of the brain. And so when you change the amount of anesthesia, that doesn't necessarily support the underlying state of the brain. It's just an indicator as a readout. So one theory of burst suppression says that burst suppression occurs when ATP runs low due to the interaction between these neural dynamics and brain metabolism. So we thought, well, these ketones increase ATP levels, so they could be neuroprotective during anesthesia by providing this additional energy source, decreasing oxidative stress, and decreasing inflammation. So we took animals and fed them a ketogenic diet so that they had ketones present in their blood compared to animals on a standard diet. And we found that these animals on the ketogenic diet had less burst suppression. So this is the burst suppression ratio. You can see that there's much more in animals on a standard diet compared to animals on the ketogenic diet. And this is just the pattern of burst suppression that we saw earlier. If you quantify this over multiple animals, you see that there is actually a significant decrease in the burst suppression ratio, and it takes longer for animals to be anesthetized. There's no difference in the time to recovery. So what this suggests is that the presence of ketones could be neuroprotective and have the potential to decrease postoperative delirium. And so now we're looking at behavioral measures in these rodents and testing that both in rodents and hoping to start a human study being able to do this. One of the nice things about ketones is that there are already ways of implementing ketones in humans, so it's relatively easy to go straight from the bench to the bedside. I'll just give a brief overview of the last two parts of my research, focusing on Alzheimer's disease and then mental health. So metabolic and sleep interventions could be useful to treat Alzheimer's disease. Why is that? Well, there's this glucose hypometabolism and sleep disruption that are early indicators or biomarkers of Alzheimer's disease. Ketones have shown promise in both animal models and human Alzheimer's disease patients. And so we are interested in looking at the energy dysfunction in Alzheimer's disease animal models and how disrupted or enhanced sleep can affect this Alzheimer's disease progression. Now, metabolism and sleep are also really important for psychiatric disorders. This is something I hadn't appreciated until the last few years, but there's a book called Brain Energy that I highly recommend that really goes into the details of this if you want to learn more information about it. Bipolar disorder and schizophrenia are serious psychiatric conditions that cause a significant reduction in the quality of life and shortened life expectancy. While their exact pathophysiology is unclear, there's increasing evidence for the role of oxidative stress and redox dysregulation as underlying mechanisms. So sleep is a critical regulator of this metabolic homeostasis and oxidative stress, and disruption of sleep and circadian rhythms contributes to the onset and the progression of bipolar disorder and schizophrenia. And interestingly, new studies have come out showing that the ketogenic diet can improve bipolar disorder, schizophrenia, depression, anxiety, and anorexia. And more studies are being done all the time. But this is a really promising area for improving mental health treatment. So therefore, we think that sleep metabolism and mental health are intricately connected and have real potential for improving the lives of people with mental disorders. So we are working on modulating sleep and measuring changes in mood in animal models of bipolar disorder. And in addition, this is very readily translatable to humans because you can increase ketones in humans through diet or fasting or exogenous ketones. And so we're building collaborations with our clinical colleagues to be able to look at the changes in sleep and circadian rhythms in patients using the ketogenic diet to improve bipolar disorder, schizophrenia, depression. So these data create rich data sets of sleep patterns and metabolites across young and old rodents, animal models of Alzheimer's disease, and psychiatric disorders, so that we'll be able to compare these similarities across multiple conditions. And having that in the database will be very helpful across multiple different researchers. Improving our understanding of the brain metabolism across these conditions has broad impacts, since so many brain diseases have components of metabolic dysregulation. These are also readily translatable to humans, as I mentioned. We're working on developing a ketone intervention for delirium and for psychiatric disorders. So these findings could lead to new metabolic therapeutics to target sleep with aging, postoperative delirium, slow progression of Alzheimer's disease, and improve mental health. And with that, I would like to thank my lab that makes this all possible. I have a number of postdocs here and summer students and past lab members, collaborators, Dr. Patrick Purdon and Zhongkang Zi, and my funding sources as well from NIH. And this is a photo of my lab. We're in Charlestown, just outside of Boston. You can see the Boston skyline in the background. And I'd like to thank everybody for your time. Thank you.